Embryonic stem cell research offers both great promise and great peril, so I have decided we must proceed with great care.
What you just heard was George Bush, former president of the United States, addressing the nation on live television about embryonic stem cells. This happened in 2001, and what prompted George Bush to address the nation on this particular day was the successful isolation of embryonic stem cells. Now, although this discovery could potentially be one of the largest scientific achievements in all of human history, George Bush and his administration were afraid of the potential ethical and moral ramifications something like this could represent for the United States and the human race as a whole. On that day, George Bush decided to effectively ban the harvesting of any new embryonic stem cells, but to continue to allow any private company that already had possession of them to continue their research and to continue advancing the field. The man I interviewed today was the man behind the company that first isolated these embryonic stem cells. The man whose company and their discoveries prompted the President of the United States to address the nation on live television all the way back in 2001. The CEO of AgeX Therapeutics, the godfather of human regeneration, Mike West. Mike, I am so excited to have you on the podcast today. I've spent the past few weeks doing my due diligence, doing my research on AgeX Therapeutics and what you guys are working on. And all I can say is that the audience, and myself included, are in for a real treat today.
Well, that's a kind thing for you to say. I'm looking forward to the discussion as well. There's not really a more exciting field to be in right now, I think, than the biotechnology of aging. It's a problem. Some people call it a disease, but it's something that all of us have, so everyone can relate to it.
Yeah, and I keep telling my friends, I keep telling basically everyone that will listen that the next 10 to 15 years, 20 years at maximum, what it means to be human is going to be completely different from a cultural perspective. This anti-aging stuff is going to change everything. And I think that AgeX is going to be a big part. AgeX and all of the stuff that you guys are working on is going to be a big part of that change. So before we dive too deep into the weeds here, I think it'd be a good idea for the audience if we just create a base layer of knowledge about the topics that we're going to be discussing today. In particular, stem cells, pluripotent stem cells, telomerase, and how these all kind of work together.
Well, can I kind of frame it in terms of the birds and the bees, if we don't mind? I won't talk about sex, only peripherally, but the birds and the bees. So reproduction, I think, is the key to understanding aging. And some people, you may have heard this idea. You remember the book, The Selfish Gene, that Dawkins wrote some years ago? He proposed that, the human body, human biology, is really just a reflection of selfish DNA wanting to replicate itself. And that's similar to the birds and the bee story that I want to relate right now. So if you're a young aging researcher, as I was when I entered the field, I was 27 or something, older or younger than I am now anyway. And where do you begin? It's sort of like Sherlock Holmes encountering a murder scene. Where do you begin to unravel the mystery of what's going on? What causes us to age, and how can we find a way to intervene in it? And what are the first clues I picked up on in this sort of detective hunt? Well, it relates to the birds and the bees. So, and if you think about it, life doesn't come from nothing. You remember we were taught that in basic biology in high school, right? Remember the idea of spontaneous generation? People used to think if you leave some rotten fruit outside, the banana turns into a fly, spontaneous generation of life. And of course, then people figured out, no, it's the fly lays an egg on the rotten banana and it becomes a a larva, and then eventually a fly. And life comes from life, they say. And cells come from cells. We're made of cells, trillions of little living entities. They're all alive. Life on Earth originally was just single cells floating around in an ocean, and they kind of stuck together and eventually evolved to a human being. Well, they're all alive. Life comes from life. Cells come from cells. So if you connected the dots of all of the trillions of cells in your body and went back in time to when you were developing in your mother's womb, you'd come down to one cell, and that was the fertilized egg, where the sperm and the egg get together. That's as much sex talk as I'll go into. If you go back before that, those were cells in that person's parents, and you do connect the dots back in time. Those were once a single cell, or there were their parents and their parents before them. I think you see where I'm headed. There's a lineage going back in time of cells that have been replicating and making humans for hundreds and hundreds of millions of years in an uninterrupted fashion, because life always comes from life. Cells comes from cells. So where I began in my search, you mentioned stem cells, was to understand sort of the root of human life that branches out to make all these stem cells in the body. How does life perpetuate itself indefinitely? And then the big question mark, why is it that the cells in our body, which we call somatic cells, or means soma in Greek, means body, why is it that the somatic cells in our body inevitably age and die in just a few decades, whereas the cells that made us have been proliferating for hundreds of millions of years? And I thought that by contrasting those two types of cells, the reproductive lineage we call the germ line, and then all the cells in our body that make up a human being, our loved ones, the brain cells and the blood cells and the bone cells, all of those are mortal and have a finite lifespan. And that's where I began, trying to understand why it is that somatic cells age, and can we somehow understand how the germline doesn't, and by comparing the two, find a way to transfer the immortality of the germline into the soma to try to stop aging, extend human lifespan, potentially to abolish aging itself.
Right. And so that is where telomerase comes in.
Right. So that was chapter 1. So I was a researcher studying cell aging. So if you take some skin cells from your arm or something, you can grow them in the laboratory dish. You feed there's this like red Kool-Aid you put on top of them, and they drink the Kool-Aid and replicate like these little pond water animals, but they're your skin cells, and they replicate. One becomes 2, 2 becomes 4, and then you can move them to a new dish, and they'll replicate. And what a scientist named Leonard Hayflick discovered in the 1960s, that recently, was that human cells of all different kinds in our body, again, the somatic cells, have a finite ability to replicate. And we call that now, in honor of him and his discovery, we call it the Hayflick Limit, or, cellular aging. And so I was studying cellular aging as a graduate student, and later on when I was in medical school. And I thought I knew what was causing it. I thought it was the telomere. It was a theory first proposed by a wild-eyed Russian theoretician named Alexei Alubnikov. He just dreamed this idea up that the ends of our DNA strands were wearing away, sort of like a fuse burning or a candle wick burning. Every time a cell divides, the end of the DNA strand was getting shorter and shorter. And that was a clocking mechanism. And that region of the telomere is called, the DNA is called the telomere, means end part in Greek. It's the end part of the DNA strands. It's kind of like the plastic on shoelaces, it's the end of the DNA. And he proposed that telomeres were shortening in somatic cells, but then this immortal germline that keeps making babies young, that there was a special gene that was an immortalizing gene. And it came to be called telomerase because it makes telomeres, and it winds this clock up. And my first company I started, like you, being an entrepreneur, I started a company called Geron, and at Geron, we spent a lot of money and a lot of effort. A lot of scientists worked very hard, and we found the gene, telomerase. And interestingly, the fun little angle here is the first human cells ever to be tested to see if this gene worked, a little bit of drama, was Leonard Hayflick's own skin cells. I happened to have his skin cells growing in the dish just when we had this gene, and we put the gene on his cells, and I called the experiment in my notebook, which I no longer have. At the top of the page, I wrote the immortalization of Dr. Hayflick, because I was confident that it would work, and it did.
And that gene is telomerase?
It's the immortalizing gene. If the gene is on, cells don't age. But it's turned off in our bone cells, our cartilage cells, our blood cells, our brain cells, all throughout the body, the soma. So as we live and cells divide in our lifetime, a clock is ticking and all of our cells are careening toward the Hayflick limit. And it's a brick wall. It stops people from living forever, the lack of telomerase.
Mike, I think you have a really good talent for explaining things in super easy to understand terms. I guess that's a skill you kind of have to have if you're the CEO of a New York Stock Exchange traded company. But if I may, I'd like to just kind of go over everything that you've just said and kind of re-explain it the way that I'm understanding it in my own terms. So originally you saw these two germlines, the sperm and the egg.
Yeah, they're germline cells, right?
Yeah.
It's a lineage of cells, yeah.
And you observed that these germline cells, they were different from the other cells in the body. These cells seemingly could replicate forever. They had no Hayflick limit. And for those that are unaware, the typical human cell can replicate about 50 times.
Plus or minus.
And if I'm not mistaken, once a cell reaches that Hayflick limit, that's when it becomes a senescent cell.
That's right. A senescent cell is a cell that's it's at the Hayflick limit. It can no longer divide, right?
So back in the 90s at Geron, which is a company that you started.
Yeah, I was the founder.
You guys were the first company to isolate the telomerase gene, which essentially was a gene that immortalizes cells. And through that discovery, you took your buddy Leonard Hayflick's cells, you edited the genomes so that they would start expressing telomerase, and essentially you created the immortalization of Leonard Hayflick, the immortalization of his cells. And through immortalizing his cells, you terminated the Hayflick limit. Well,
the joke was, if you go into my YouTube channel, I've actually got an interview with Len, or we're recounting the story, and he just happened to be in the company and donated a little piece of skin for reasons I explain in that video. I won't recount here. But I was a joke. I thought, he said, you want some of my skin? And I said, well, sure, because I want to measure the Hayflick limit, the true Hayflick limit. How many doublings your skin will go? We always talk about the Hayflick limit. Let's get the actual limit of Len Hayflick cells. And it just so happened, though, that it was just at that time when we had the gene cloned. And, I have a lot of respect for Len. He's still alive, still very active. And I felt, he deserves the honor of his cells being the first to be immortalized by the hand of man, so to speak. And so his cells were the first to see, the expression of telomerase through human intervention, right?
That's a great story, and I'm sure that was quite an honor for him.
He enjoyed the experiment, but experiments normally don't work. So at the time, I was dating one of the scientists that, I think played the most important role in finding the gene. Her name was Karen Chapman, and I'm married to her now. And we went out for dinner that night and had some wine. And she said, so tell me the truth, kind of grinning, do you think that experiment's actually going to work? And I know what she was getting at. In science, they call it research because you search and you search again and you search again. Experiments in science is full of failure and testing of hypotheses that don't work. And the chance that one gene, it could have been 6 genes, right? That one gene is turned off in somatic cells, and adding that one gene back would stop the aging of cells. Len himself didn't believe it would work. He thought that there were hundreds. Len thought the aging of cells was wear and tear. It was just like entropy. Things fall apart. Your car falls apart. The tires wear out on your car. The cells get old and tired and worn out. And that was the prevailing hypothesis. And what we were saying at Geron was, well, not really. There's a gene that is turned off very early in life, and then there's a clock ticking in every cell except for these germline cells that make babies. And and then time bombs are going off as we get older, and those senescent cells cause our aging and death. And the idea that one gene was the sole cause, I agree that Karen was right to be skeptical, but I won the bet. She said, when pigs fly, and I've got a flying pig trophy on my desk today at work. Sometimes pigs do fly.
That is a very lovely story, Mike, and even a bit romantic, the story behind the isolation of the telomerase gene. Which brings me to the next thing that I really wanted to bring up, because I saw in one of your interviews, or I saw it on the website somewhere, that I thought this was super interesting. For those that don't know, cancer cells are essentially immortal cells. That's what the problem is behind cancer cells, is that they keep replicating forever. They keep replicating cancer cells. And one of the interesting things that you guys discovered about cancer cells is that they actually have the telomerase gene turned back on.
Yeah. So there's at least one science fiction novel. It might be The Sixth Commandment. I'm not sure. I haven't read that story in many years. But it was about a gerontologist who was doing research on aging. This is many years before the Geron was started and so on. And there was an old folks home next to the research facility, and some insurance person was coming to investigate because these older patients were all getting peculiar cancers. Turns out the scientist had, in this story, had found the immortality gene and was testing it on people and causing cancer. Well, what was that all about? Cancer cells, when they're grown in the laboratory dish, typically do not age. So we came to call those cells immortal cells. It's a scientific term. It's not meant to imply any theological concept. It's, we're not talking about the immortality of the soul or something. We're just saying that the cells don't age. They don't stop dividing. Normal cells stop dividing and age. There's never been, to my knowledge, a human normal cell, other than if we get to talk about pluripotent stem cells, cultured germline cells, blood cells, brain cells, any kind of cell from the body, all have this Hayflick phenomenon. But when cells become cancer and you put them in the dish, they no longer age. So we say that they've immortalized. And no one knew the mechanism of that. Well, that was easy for us to figure out. Even before we had the gene, we could tell if telomerase was turned on or not. And we published back in '94 that over 90% of cancers have turned telomerase on. And so was it Freddy Krueger or something? I'm not a big movie fan, but there's one movie you shoot him and he gets back up and he keeps rising from the dead or whatever. You can't kill him. Cancers are kind of like that. You hit them with radiation and chemotherapy, and they just keep coming back. And the reason is the cells will not stop dividing. You can kill some cells, but cancer cells, but they'll replicate forever. And that's what makes them so deadly. So doesn't that mean the use of telomerase would cause cancer? No, actually it doesn't. It's a long, complicated discussion, probably longer than we would want to talk about here. But I'll just say cancer is a multiple step transformation of a normal cell into what causes cancer. It's kind of like a runaway car. The accelerator is stuck down, so cells are dividing out of control. There's no braking mechanisms anymore in the cell, so they can't stop. And so the accelerator's stuck, the brake is broken in your car, and it has an infinite fuel supply, so it'll never stop running. And the infinite fuel supply is telomerase. Nature, we think, makes us mortal as a way to help prevent cancer. So if the car will run out of gas, that's one way to control a runaway car that nature uses to help reduce the chances of cancer. But if the brakes are working normally in a car and the accelerator is working normally, and you had an infinite fuel supply, that doesn't make it a runaway car. Does that make sense?
Yeah, that makes sense. And it actually answers a question that I wanted to bring up later on when we talked about induced tissue regeneration, which is something you guys are working on. Because obviously, when the thought of having cells that are immortal, that will replicate forever, you can't help but think there must be some sort of cancer risk here. But I think that your explanation is actually really great, and it's a really good analogy as well, using a car as the cell, essentially. So as long as everything else in the car is working properly, as long as the brakes are working, as long as the steering wheel is working, then as long as those are working, if you add some fuel, an unlimited fuel supply, it's not going to get out of control. And that's essentially how the cell works. As long as everything else in the cell is working properly, adding the fuel supply, aka the telomerase, to keep the cell going forever, it's not going to cause a lot of problems. It's not going to cause cancer.
Yeah. And on top of that, what almost all scientists, to a person that's in this field of aging research, believes is that telomerase is this amazing enzyme that's lacking and causing cell aging and causing a lot of human aging. But what we would do is, like a normal car, drive it into the gas station and then re-tap up the fuel tank, not give you an infinite fuel supply. We just reset telomeres and then take telomerase away. So as we say, we would transiently, temporarily express the gene to wind the clock back up in our cells to make them younger, and then take the key to that clock away. And so there's plenty, we think, of what we call it as a therapeutic window. There's plenty of opportunity to use telomerase, especially when you're very sick from an age-related disease, coronary disease or something. There's plenty of room to intervene in aging without causing cancer. And at least that's what we believe currently.
So that is a pretty good place to start. I feel like we've laid a pretty solid foundation for the audience. So let's go ahead and get started with what AgeX is working on, and let's talk about pluripotent stem cells and all the, quite frankly, crazy stuff you guys are doing with those.
Yeah, so there's 3.5 major revolutions that I've been personally involved with. One, we just kind of talked about this telomere thing. If you were in the cell aging field, and there weren't very many people in that field back when I was there, that was a absolutely unanticipated revolution in thought that cell aging was under control of one gene, and you could immortalize a cell. I mean, he would have been laughed out of court before we'd actually proved that that was true. A second one. So what occurred to me was we should be able to actually capture this germline in the dish. It makes, as I've said, babies are always born young, and we keep making people keep making babies, and then their cells make more babies, and so on, as I previously mentioned. Well, what if we could capture those magical cells, the germline, in the dish? Potentially, we could use them not to make people, but since just like babies are born young, we could use it to make just cell types that are born young. Young heart muscle for heart failure, young cartilage for your joints. When you get older, the number one complaint in aging is the pain of arthritis. It's the number one complaint. And, if we could reline the surfaces of your joint with young cartilage, that would be really amazing. If we could make young cells for your midbrain for Parkinson's disease, you know where I'm going with this, right? It goes on and on and on. And some people just would like to make hair for their head and everything else. So you talk about unmet needs in medicine. There's never been a real practical way of making young replacement cells. Your car, if it wears out, you can go to a parts store and get new spark plugs for an older car. You can get new tires put on. You can keep a car running indefinitely because you can always get parts. The human body is as important as it is to all of us. We've never had a way of rebuilding the body. And so I had this interest in trying to isolate the human germline in the dish and use it as a manufacturing platform to make young cells of any kind. And you can't use sperm and egg because they don't divide. But right after the egg is fertilized by a sperm, a cluster of cells form. It's called the inner cell mass. And it's raw material for life. And these cells, if you went in and divided that little cluster into two, you'll get identical twins. That's how they form. You can divide them again and get identical quadruplets. That even happens sometimes just naturally. It's just these cells have not yet decided to make one person or two people, or even a part of a person. Sometimes two of these will collide together and you'll get a person that's made of two different DNA types. So anyway, these early cells of the inner cell mass, I felt they were being thrown away. When you use in vitro fertilization, as is commonly done for people who have difficulty conceiving children on their own, excess fertilized egg cells were thrown away. And what I organized back in the mid-90s was a collaboration with some scientists, Roger Peterson at UCSF, James Thompson at the University of Wisconsin at Madison, and John Gearhart at Johns Hopkins Medical School. In this collaborative effort, we tried to capture these, broadly speaking, they're called pluripotent stem cells, meaning they could make the power or the potency to make everything in the body.
Sorry to interrupt you really quick. I just wanted to clarify. These stem cells you're talking about, are they the same as embryonic stem cells?
Yeah, so if you culture them from the inner cell mass, from the fertilized egg cell, they're called embryonic stem cells, right? So when President Bush introduced the country really to these cells. His first address to the country from his home was on this discovery this group that I pulled together. So it became very controversial. He said, wow, if you listen to his talk, President Bush was saying about how ethically problematic this was and, grave ethical dilemma, but he felt that it could really change medicine. And he said, okay, so the ones that were already made, he would allow federal funds to be used to, use them in medicine. I don't know if you remember all that debate. It was a big debate. And so that became a kind of a national debate about these cells. But the scientific community realized for the first time, this is the first time we ever had the human germline in the dish, and you could study all of human development, the origination of heart tissue, of the central nervous system, kidney development. It opened the door to just an enormous new field of medicine. Early on, before this was all public, I was presenting these ideas in a meeting, and a scientist named Bill Hazeltine, who used to be the CEO of Human Genome Sciences, said this field needs a name. It should be called regenerative medicine. And that name stuck. So now it's a whole field of medicine, regenerative medicine. The concept is now we have a pathway to make young cells of any kind to repair any tissue in the body as a part supply store for the human body. We actually know how to do that now. And, a lot of these products are not yet available because it takes time to get FDA approval, but it's a major area of research now for scientists around the world. So, it's a strategy to deal with aging. It's a tool in the toolbox, young cells for old people to repair the human body.
Let me ask you a question.
Yep.
With these pluripotent stem cells, can these be used to grow entire organs, a replacement heart?
Yeah, they can. So they're so early in development. I mean, they know on their own, they know how to form a human body. And so they don't make people or in a dish, but they make tissues and they self-organize. And so you'll see beating heart muscle in the dish. And people have even seen, complex tissues like kidney tubules forming and so on. Yeah, they very unusual in that respect. They're capable of self-organizing.
So I just want to say for the audience, if you're not following along, these pluripotent stem cells can literally create new organs on their own, whether it's a heart muscle or I would say a liver, or really it can turn into any cell that the human body has. That is absolutely incredible.
Well, it's more incredible than that because, as you point out, they can, organize into complex tissues, which cells and tissues from the adult human cannot do that anymore. But they have telomerase. Yeah, pluripotent stem cells, because they, as far as they're concerned, they could still become a sperm and an egg. And so they haven't committed themselves to mortality at that point. So because they're the first, and to my knowledge only, normal, I have to say normal because cancer cells are immortal, but they're the first and only human normal cell ever isolated that's naturally immortal. That's why I wrote a book about some of this I called The Immortal Cell, because the quest was to find the naturally immortal cell, and we found it with the pluripotent cell. It is the human germline. But now, why is that important? It's not just a scientific curiosity. If the cell will replicate indefinitely, it opens the door for scientists to go in and, I wanted to say tinker with the DNA. That's not the right word because people will say, scientists are playing games. It's not really. You can make designed changes in the DNA that are designed to make supercharged cells or to correct genetic defects in the cell. But you can go in, just like you do in a document on a computer. You know how you can do find and replace in a Word document? If you were going to send me a letter and it was written to Bob, you can say, find Bob and replace it with Mike. And with a keystroke, all throughout the document, it would find and replace. There's techniques like that that we have in medical research. If we have a cell that will replicate indefinitely, it allows us to do find and replace. So we can take the DNA and design it any way we want. I mean that literally. We can now make any kind of human cell genetically engineered in any way that's a really powerful technology platform.
That is, it's like you're reading a page from a science fiction book that I would never believe. So let me just clarify again for those that may not fully comprehend what you're saying. Basically, just for let's just say the heart. Okay, let's say you have had heart problems your whole life. With these pluripotent stem cells, not only could they give new cells to your heart, potentially replace all the broken parts, they could genetically modify it so that now you have a better heart than normal. Now you have a heart that works significantly better than normal. Maybe can I go as far as saying a heart of a super athlete?
Yeah, well, there's some genes. The problem is evolution, if we're allowed to proceed for hundreds of millions of years, presumably could turn the human species into something superhuman. And what those changes are, it's hard for scientists to know exactly how to, again, I want to say it's word tinker, but, we kind of are tinkering because the DNA, the code of life that makes us from a single cell and makes us who we are, gives us our eye color and everything else. Some of the genes are very easy to understand, but the coding language itself is really complicated. It's like going into a computer code for Microsoft Word again or something, if I use that analogy, and trying to change the program when we didn't write the original program. And it's tough. But you mentioned the super muscle thing. There's one gene that we know, it's called myostatin. If you go in and just take the gene away, we call it a knockout. If you go in and just remove the gene, you get double muscling, it's called. You get up to 6 times the muscle content in an animal that you normally would have, and with no exercise. It's not like the animal exercises more or something. It's just the resting muscle mass. So, we know how to make Arnold Schwarzenegger babies that never have to exercise. That's just an example of what could be done today.
Mike, me and a lot of my friends were very into fitness, very into working out. So you saying that, it's music to my ears. And I know that there's going to be a lot of people out there that are interested in that.
I'm not advocating it. And I'm just saying that, it's reality. It's, there's publications where for years we've had the mouse embryonic stem cell long before we had the human. And this gene has been, knocked out, as it said, in mouse. And you can see these mice, there's published papers on this double muscle phenotype. And they have up to 6 times the muscle content of a normal mouse. They look like little mini mice.
And so you just have these jacked mice walking around, these alpha male mice in the group.
Yeah.
Wow.
Yeah, and we know it works in not just mice, because there are rare, cows that have this incredibly muscular phenotype. It's just kind of ridiculous to look at. They've seen the muscles bulging, and they've been sequenced, and we know it's the myostatin gene and so on. But that's just one example. I mean, a lot of people point to, because this is a doorway to introduce genetic modifications, people want to make dogs that glow in the dark for a pet, and all kinds of crazy things. There's a lot of things that science knows how to do. The question is, what do we do with all these powerful technologies? I'm really an advocate of the medical uses of the technologies to alleviate human suffering, rather than just tinkering life with life for the sake of tinkering.
I think you're 100% correct. Yeah. And I mean, going forward, we need to use these powers for good, for sure. But you can't help but just be excited about what the other possible potentials are out there.
Yeah, it's, I don't even know where to begin. The power of modern medical research is very quickly paced faster, than what even the wild-eyed science fiction writer would have written a few years ago.
So, wow, I'm blown away. My mind is absolutely blown right now. I want to ask you a little bit about some of what AgeX is working on, some of your guys' products. PureStem in particular, although we've kind of gone over exactly what it does already, But Pure Stem is basically what we've talked about, which is growing any type of cell from the pluripotent stem cells, repair. And you guys are focusing on brown adipose tissue and vascular something.
Yeah. So a big problem in aging is metabolic imbalances. We gain weight, central obesity, type 2 diabetes. We think that's due in large part to a loss of a anti-fat cell, really. It's called a fat cell, a brown fat cell, but it's actually works in reverse. It pulls fat out of the circulation and burns it to generate heat. They're called brown adipocytes. And we know how to make those. Remember, pluripotent cells make everything. We figured out how to steer them into making just that cell type and just vascular forming cells for like your heart. The heart attack is due to the lack of blood flow to the heart. And we have some cells that can, rebuild a vascular tree to the heart or other tissues like that and need increased blood flow. So, we're trying to implement, looking back in time, some of my previous companies, I have cells in human clinical trials even today for spinal cord injury and macular degeneration, the leading cause of blindness and aging. So, we're getting some of these products into human clinical trials, and hopefully, soon, some of them will be approved for human application. But it's really just the first halting steps, really, in this field we call regenerative medicine.
Yeah, I did see on your website the brown adipose tissue and the vascular cells that you guys are working on are in preclinical trials. And what exactly does that mean, preclinical trials?
FDA is first and foremost concerned about safety. Cells, the transplantation of cells is historically been very safe, far more safe than the use of drugs, for instance. About 9 out of 10 drugs fail in clinical trials. Cells transplantations normally are safe, but FDA really is concerned about safety. It's their number one concern. So we have to demonstrate in animals that the transplantation of these cells doesn't cause some kind of a health problem. And those studies are what we call preclinical studies. So they're typically animal-based or sometimes based on laboratory experiments, but they're all designed to demonstrate to the FDA that the product should be safe to at least begin a safety trial in humans and to prove that they're safe. And then you go to the efficacy to see, do they actually benefit the patient?
Well, I've got a few questions about that, but if you can speak to it, what are your results so far with the animal trials? I mean, are there any results you can publicly reveal at this time?
Yeah, we're a public company, so we don't only disclose that which we've published on or somehow announced. What I can say is that there's evidence, considerable amount of evidence for both cell types that they're beneficial. Work out of the Harvard has a thing called the Joslin Diabetes Center, and with age, you lose this brown fat, and it leads to what we believe is this accumulation of fat around your waist, and then insulin insensitivity, or so adult onset diabetes. That's a real problem, by the way, not just obesity, but type 2 diabetes. It has a lot of bad side effects, including heart disease.
As humans, at what age do we typically start to lose a significant amount of that brown adipose tissue?
Yeah, it's kind of linear, it's progressive with time. So we have a lot of it when we're born. And some people believe that mammals, when they're born, I don't know if you've ever seen little pups, dogs or mice, they're called pups as well. They don't have much hair on them. They're kind of like these little skinny little things. And when mom leaves, they could quickly die just from being cold, especially in colder climates. So animals, including humans, when we're first born, we have a lot of this brown adipose tissue in our chest region. And we get cold, and it's just like a thermostat that turns on your heater. When you feel cold, the brain sends signal to the brown fat, and it literally generates heat. It's like a furnace in the body. But you lose that as you get older. And the result is Without this tissue, you cannot absorb glucose and circulating fat the way you could when you were younger. And so, your glucose can go out of control, your circulating glucose levels, and fat can start accumulating like in your coronary arteries. So really depositing fat in inappropriate place. That's what a coronary disease is. Cholesterol we hear about.
Right. That makes sense. And so once you guys, and hopefully if you guys get all the way through clinical trials with your targeting the brown adipose tissue, targeting the vascular stuff, how easy is it to then use that same technology and get things through, let's say, heart tissue?
So with the first product ever made from these cells were cells for spinal cord injury. That was done at my first company, Geron. It was the largest, most extensive preclinical package ever presented to the FDA in the history of the FDA. It was so much work was required. It was a really heroic effort by the Geron team. And but every new product we approached the FDA on got easier and easier and easier. So now these new products they're still, they still take years and a lot of capital to be developed, but, applying this to regenerating the heart or for Parkinson's and so on is, relatively straightforward. It's not unlike drug development at this point.
That's great news. And for the induced tissue regeneration, now that's the telomerase part of your company, correct?
Well, you said you were kind of amazed by the science. Now I'm going to lose all you and your listeners because you're going to say, no, I was kind of going along with this guy, this is kind of neat stuff. And I Googled telomeres and telomerase and wow, some people are, our old collaborators got Nobel Prizes for it. Liz Blackburn and Carol Greider, stuff like that. Yeah, wow, that looks real. Yeah, Nobel Committee thought so. Yeah. Then I'm going to tell you about ITR, and you say, okay, so this guy went off the rails. Let me tell you the ITR story. Let me tell you the story. It's not a story. It's real.
I'm excited.
But it's new. And you're not going to find a lot about it online. There was actually just an article written in the New York Times a few days ago. I think Nicholas Wade wrote it. And he cited Lenny Guarente at MIT saying, this is a big deal. So, it's starting to get out there. So what's the deal? I began with Dolly the sheep. Back to the birds and the bees here. So normally life begins with a fertilized egg, as we said. Well, turns out something that no one thought would ever work. If you take a cell from the body, a somatic cell that ages and is immortal, right? It's differentiated too. It's now a skin cell or a heart cell or whatever. If you take that cell and its DNA, its little blueprint of life, and you put it into an egg cell, the egg cell will act like a time machine. And it reverses the aging of that somatic cell and takes it back to an embryonic state. And if you put that cell into a uterus, you can generate an animal identical to where that cell came from. So that's cloning, of course, somatic cell nuclear transfer, SCNT. So Dolly was cloned from a cell from breast tissue. They named her after Dolly Parton. It's a typical humor of a overworked scientist. So what was once a breast epithelial cell was put into an egg cell, and the cell was taken back in time, and a young lamb was born. And I was really entranced I thought that this was reversing aging itself in a cell. I bought the U.S. cloning effort. It was a company called Advanced Cell Technology and an animal cloning company. My old company, Geron, which I had left, bought the Dolly Roslin company. And they published in Nature, this prestigious magazine, that it didn't reverse aging. So Dolly was indeed a lamb, but she was a sheep in lamb's clothing. She was actually born old. And a lot of people believe that. That's kind of a myth out there. We published a year later, in the year 2000, in Science, another kind of competing journal, that in cows at least, somatic cell nuclear transfer, when you take an aged cell from the body and you put it into an egg cell, the egg cell acts like a time machine, and we showed that it reset telomeres, it activated telomerase and rewound the clock, and the resulting baby cow was born young, totally normally young. Now, why do I tell you that story? I thought that was truly magic on multiple dimensions. One, that the process of making a body, that arrow we call it development, could be turned into reverse. I mean, why would it go? It never goes in reverse in nature. How could that possibly be true? And secondly, that something in the egg cell reverses aging in the truest of sense. I had to study this. And so beginning around 88, 89, that's all I worked on for years and years and years. And we published in 2001 the first, right around the time that Bush had approved, talked about the embryonic stem cells, we published the human nuclear transfer, taking a human cell in a ride and a human egg cell, a human time machine. That was a little controversial. President Bush vehemently disagreed with us, ordered the patent office to remove one of our patents after it issued. And we were kind of at war with President Bush on that issue. Our intent, I think, was misunderstood. Why not was. We weren't trying to clone people. We were trying to clone cells. And I wanted to find a way to reverse the aging of cells. Well, over the years, we found the molecules in the egg cell that are like telomerase, that is capable of immortalizing a cell. We found the genes, in this case it was several genes, that are capable of acting like a time machine. So just like if you put telomerase into Len Hayflick's cell, it can extend the telomeres of a cell and immortalize the cells. By putting these ITR genes, we call it induced tissue regeneration, ITR, it can take cells back in time. And what we're doing is developing it to take cells back in time in the human body. So we're working on delivering telomerase and these reprogramming genes, as we call them, into the human body to take your cells literally back in time, to reverse the aging of cells and tissues in the body in the truest and fullest sense of the word. Is that possible? Well, we haven't published much of our data. We published some at AgeX, we published some basic science, but we have two competitors in the field, friendly competitors, I would say. One is Turn, T-U-R-N, like turning back the clock. They're a group out of Stanford. They just published this paper, which was written up in the New York Times. And they're seeing that these genes reverse the aging of tissues of various kinds. The other competing effort is David Sinclair's group at Harvard Medical School. And they published that when you put these genes into the eye, you can reverse the aging of the eye and induce a characteristic that cells and tissues have when you're very young, when the body's first forming, to naturally repair itself in a very profound way. And so that's why I call it induced tissue regeneration, or ITR. This technology is the most profound science I've ever encountered in my life. And I believe it's going to be really history in the making. It's what's no one's expecting to happen. I think no one thought we'd ever reverse the aging of cells with telomeres or cloning, that you could actually clone an animal, but it actually reversed the aging process. I mean, literally, in the body of an animal or a human. Aging researchers never thought that was possible. Slow aging, sure. We knew we could do that. Extend human lifespan or animal lifespan, we've known we could do that for years, 30% or something. But reverse the aging of an animal or a tissue, that's an eye-opener. I'm sure the average listener will think, oh, that just simply can't be true, but it is true. And that's what the public will be hearing about, I'm sure, over the next few years as papers are published.
And so let me ask you to put the significance of this into practical terms so people can really wrap their mind around it. When you say reversing our cells, reversing the aging of our cells, I mean, what exactly does that mean? Are we saying that with this therapy, I'll be able to go back to being a child? Or what exactly do you mean by this?
Benjamin Button? Yeah, right. No, so, well, I guess we don't know because we haven't done it in a human, but I wouldn't think so. The growth pathways appear to us in all the modern medical technologies are incredibly powerful, with this RNA sequencing and everything. It's just so much data. And it appears that the mechanisms that control the size of the human body are separate mechanisms from those that are controlling aging. At one point, some scientists thought maybe they were the same, and they thought that aging was sort of a mechanism to prevent us from becoming giants. We stopped growing when we reached adulthood, and that same process was causing us to age. I don't think that's true, and I think most chiropractors don't believe that that's true. So if we reverse the aging of a tissue, we don't think you would literally get smaller and smaller and become a baby. What we think is what would happen is that the way we're designing this technology is that you would induce molecular mechanisms that really you don't even have when you're born, an intrinsic ability to repair your body scarlessly, like in a heart attack. Early in development, you have a heart attack, the body just regrows new heart muscle. And we believe we can reawaken that and reverse and add back telomere length through the use of telomerase, but allow the body to kill off old cells naturally and replace them with young, healthy cells and rebuild the body in a healthy state, but without regressing size and development. We think that's a pathway commonly called the hippo pathway, like the hippopotamus that controls body size. We think it's different. So, but how all this would work is this is entirely new technology, and right off the lab bench, I'm just kind of giving you a glimpse where the science is at the moment, and the current animal data looks extraordinary, and the science is very strong, and we're quite excited about it.
I'm excited about it as well, Mike. This is really, really cool stuff. So let me make sure that I'm following along with everything that you said so far correctly. Basically, I mean, it's not going to be Benjamin Button, not likely, but but what it is going to be is, if we have chronic injuries, if we have a heart attack, I mean, anything like that happens with this induced tissue regeneration, it will be able to heal you 100%.
That's what we're aiming at. I think it's possible. We had that. Let me put it this way. We know it's well established that we can reverse the aging of your cells all the way back to the beginning of life, right? You know that, right? That's called induced, not tissue regeneration, but induced pluripotency, IPS cells. So I can take your cells back to day 6 of development and then make beating heart muscle in the dish. And it's your heart muscle that you had in the first few weeks when your body was forming. We know that. That's well-established science.
Sorry, but that is also insane.
Yeah.
It's just so cool. But go ahead.
I know it's insane. It's cloning, but I used to call it analytical, cloning. It's cloning without using the egg cell. It's using molecules that do all the work. And it's called induced pluripotent. So we know that we can reverse time zero and truly make all these age markers go in reverse and take you all the way back to the cells you were born from. Now, if I did that in you, you would just be this green, pile of cells. You'd lose all of your differentiated cells. You'd be slime, and that's not a good thing. So instead, what I'm describing here is taking the train ride instead of back in time all the way back to the beginning of life, jumping off the train partway. The majority of scientists call it partial reprogramming, and that's the concept. You're taking this miracle of reprogramming, of taking a cell back to the beginning of life, but you're only taking it part way and you're jumping off the train. And like I said, we all had, when our bodies were first forming, we had this amazing ability to repair a tissue. If you go into a baby that's just forming in its mother's womb and you cut the skin, in 72 hours, the skin's completely normal in appearance, scarlessly. And if you took a chunk out of their heart or induced a heart attack, and you of course do this in a mouse or something, not a human, it can regenerate. The spinal cord can regenerate in humans. We know that from some data. So what we're interested in doing is taking the cells in your body back to that early stage and then letting them go back and forward in time again, and also resetting telomere length. Why am I optimistic about the outcome? Because nature shows us that such a reactivation of regeneration and telomeres can thwart aging. So some animals can regenerate profoundly and have telomerase all the time, and they don't get cancer, and they don't age. So, these little planaria, you've seen these little flatworms, right? They have two buggy eyes and crawl around. You can cut their head off, and their tail will grow a new head, and their head will grow a new tail, and you can do that forever. They're said to be immortal under the edge of the knife. And they can regenerate, they have telomerase, and they just keep going, and there's no aging in those animals. And there's more complex animals. There's an animal in, I think it's in Slovenia, called the Olm, O-L-M, and it can regenerate and presumably has telomerase. So I haven't seen definitive data yet on that. We know it can live at least 100 years, and most people think it's an immortal vertebrate animal. So the ability to regenerate and for our cells to replicate without a Hayflick limit, we think are the two most profound means to intervene in aging been found to date. That isn't to say, there's other strategies, there may be, but animals that have the ability to regenerate and can do it endlessly typically don't age. So regardless, these will be very important in medicine. The ability to regrow a heart after a heart attack and the ability to induce that would be all by itself, a very important advance, of course.
Yeah, and one thing that I saw in, again, one of your videos, and all of these videos that I've been watching, I'll include them in the podcast show notes for anyone that's interested, because there's some really, really cool stuff in the AgeX Therapeutics YouTube channel. But one thing that I saw, I think it was in your Undoing Aging talk, was that you mentioned that all of these mechanisms of aging that a lot of people are targeting and trying to tweak and figure out, mTOR, AMPK, what your theory is, what the AgeX theory is that really the reason why those are so beneficial for humans is because they ultimately enable tissue regeneration, which is exactly what you're working on.
Yeah. Yeah, we believe that that's true. And what I think researchers will be quick to point out, Kathleen Kenyon wrote an article in Nature Genetics, I think, Nature BioTech, maybe it was. And she was talking about, she's at Calico now, Google's anti-aging company. And in this article, she was talking about now scientists, by studying animals like these worms, C. elegans, and so on, they found genes that extend lifespan. As I said, that's old hat. Gerontologists have been extending the lifespan of animals for years, and we think that provides important clues about aging. And we found these genes, the growth hormone pathways, mTOR you mentioned, and so on. But in this article, she says, but still what we don't know is why, tinkering again with those pathways, do we extend the aging lifespan of an animal? How does it actually, why does it impact aging? And that's been an unanswered question. Well, what we think is that normally nature turns off these regenerative pathways, and they can be reawakened in certain kinds of stress. So for many millions of years, we haven't had the luxury of agriculture and the wealth that we have in today's world. And there was commonly, for all animals, feast-famine cycles, right? There'd be a long summer, winter, whatever, and we wouldn't have enough to eat. And so what the body does in those situations is it starts sort of catabolizing itself. First you burn the fat, of course, when you're starving, and you lose, that's what fat is. It means storing energy for feast-famine. And then you'll start sort of doing the same with muscle. You actually start losing muscle mass. You're kind of eating your own muscle in a sense. But let's say that we go through that famine cycle, and now it's springtime and there's abundant fish and fruit and everything else. You chow down, that's the feast part of the cycle. But the body needs to be able to regenerate the tissue that was used up in the famine. And so nature kind of turns that back on. It turns on regeneration a bit to allow you to rebuild your muscle mass again. And of course, fat easily grows. That's never been a problem for humans.
Unfortunately.
And so if you restrict the dietary intake of a human and most animals, you extend a longevity. And we think it's by nature's reawakening some of these regenerative pathways. And it's kind of complex biology. But so what we're doing, we believe in the gerontology community in these meetings, people working on the different aspects of aging are kind of like the proverbial story of blindfolded people touching an elephant, and some have a hold of the tail and some have a hold of the trunk. They say it's like a rope. No, it's like a trunk of a tree. We're all looking at different aspects of aging, but we're comparing our notes or lifting the blindfolds and realizing actually there's a unified theory of aging here, and we're all studying the same thing. And we're starting to put the puzzle pieces together to a really profound understanding of how we age, and then, of course, powerful tools to really get at the central mechanisms and point the arrow in reverse.
When I first got into this whole anti-aging thing, basically I saw David Sinclair on Joe Rogan, and I kind of went through as I would call it, the David Sinclair protocol. I bought a bunch of NAD boosters. I started using the sauna, and I started fasting. And of course, all of those things are extremely good for you and healthy for you. But as I've talked to more aging experts, they really aren't going to move the needle that much. And for those that aren't aware, the pathways that I was just talking about, AMPK, IGF-1, all those aging pathways, fasting and sauna, all of that works on those pathways. But ultimately, what's really going to move the needle, because those things, from what I understand, in a best case scenario, may increase your lifespan by 3 to 5 years. Am I correct in that?
Right. Yeah, well, we don't know, of course. We're guessing based on animal data, but right, you're looking at a modest impact on longevity. Of course, yeah, there's no
Exactly.
question.
And so people come to me because, in my community, they kind of know me as I keep up to date as much as possible with this anti-aging stuff. And the thing I've learned the most is that, that stuff's not going to do much, but this stuff that's coming out in the next 5 to 10 years, that's going to move the needle. And I guess that brings me to my next question is, in a best case scenario, when would what AgeX is working on be available to the public?
I really like giving straight answers to things. I hate it when people waffle, but the true answer to that, it's entirely dependent on how many scientists are working on it. It's like, Kennedy saying we're going to go to the moon in this generation. Well, what year will it be? Will it be 1969, 1967, or will it be the 1970s? So it really depends on how many people. We have formulations in our hands. Turn has, and David Sinclair Groups has this part. They call it partial reprogramming, and I call it induced tissue regeneration. Different names for basically the same thing. And we all have formulations that could theoretically be used in humans today, but we won't. I won't anyway. Put it into a human today, and I tend to work with FDA, and we've got this is entirely new. I don't know how to stress this enough. This is a really powerful thing we're talking about. It's kind of like a atom bomb. I mean, this completely reprograms the DNA in a cell. This isn't subtle. This is a very powerful intervention. So the burden on us to prove safety is rather large. And when this will be available to humans in a clinical trial setting or to be launched as a product that can be bought by visiting your doctor is entirely dependent on how many people. And it's difficult for me to project it at this point. AgeX is a very small company, and we need pharmaceutical partners or some other means to get a lot of people to make it happen. It will happen in the lifetime of many now living, inevitably. But I wouldn't even want to try to project a year at this point.
That's fair. So could be 5 years, could be 100 years, somewhere in that timeline somehow.
No, not 800. Yeah, we're talking earliest 5 years, and with massive effort. And I would say on the outside, this will be in humans. Even at the slowest would be in 15, 20 years. We're not talking about 100 years. Because, the formulations are already in existence. It's not like a drug in medicine and in drug development. Oftentimes you have an understanding about something like cancer, and you start searching for a drug that will hit the target and potentially be used as a cancer therapy. And so that's the beginning of discovery research, where you're just searching for the agent. We're beyond that. We have things that could be used therapeutically today. So, it's a question of, more like, there's still discovery research that needs to be done for sure, but we're mostly talking about the challenges of demonstrating safety and animal, likely preclinical studies, and then the beginning of a human clinical trial.
Well, it's a ways off, but it's going to change everything.
Yeah, let me just say, it will change everything. It really will change everything. The how we think about degenerative diseases, and then of course aging, is going to forever change based on some of these discoveries we've been talking about today. It really changes the whole paradigm of how we think about aging.
I think you've done an amazing job explaining this because 90% of the audience, 99% of the audience is people who are just casually interested. And I think you've done such an amazing job today getting me excited. And I think a ton of people are going to get really excited about this. And you guys are publicly traded. Just saying, you can invest in this company right now on the New York Stock Exchange.
Since you mentioned being public, we have filings with the SEC. Anyone looking at the company should carefully review all of our filings. they carefully review them before making any investment in the company, please.
Of course, this is not investment advice, but it is a super exciting company. I want to finish things off, if I can, with just asking you a few fun questions, if you don't mind.
Oh, yeah, sure, of course.
First of all, let's really, I want to talk best case scenario. I want to talk if AgeX knocks everything out of the park with the Pure Stem, with the ITR, with everything that you guys are working on. How drastically do you see this increasing human life and health span?
For years, as a young gerontologist, it was clear to me, people in the field of aging research, it was verboten to answer such a question. And it was unspoken rule, but it was a very clear rule. I never understood that rule because it's a logical question, right? And part of it might be there's a lot of quack medicine out there, and I think I would predict here that as this science becomes more commonly known, as science papers are published and people listening to programs such as what you're putting on and so on, people will start, this become more and more knowledge. The fake medicine, quack medicine, the clinics say, come get it now, come get reprogramming now, will be everywhere. And I think there's a reticence in the aging community to raise hopes and then have people go out and spend their money with the fraudsters out there, without naming any particular group or thing. And so, on the other hand, scientists, I think, have a moral obligation to try to communicate where science is and where it's headed. I think we've largely not done well in that regard. I think the public is woefully unaware of a lot of the science going on in medical research. And so I feel a need to be, quite transparent about this. I don't believe that there's an intrinsic limit to human lifespan, that there needs to be. There certainly is now. 120 years or so is the upper limit. That's what we call lifespan. Life expectancy is when half of a group of people have died. So that's the average time people live. And you hear, it's in the 70s, differing from men and women. Lifespan, the maximum achievable lifespan, is about 120. But that's entirely a brick wall that results from this unique biology in our somatic cells. As I said, we're made of cells that have been proliferating for billions of years in the past. And then suddenly, within a few decades, boom, they'll age and die. It's a unique characteristic of the cells in our body. And we know we can reprogram a somatic cell back to the germline and reawaken the immortalizing gene, telomerase, and these regenerative pathways that allow the body to repair itself indefinitely. So I don't believe there's an intrinsic limit to human lifespan. They say actuarials that study life insurance and things say that if we eliminated the biology of aging with drugs and technologies such as we've been discussing, humans would probably live somewhere in the realm of 600 to 700 years. And in today's world, with the risks we have, on average, life expectancy would probably be about 600 to 700 years, and then we get hit by a bus or something, killed by coronavirus,
right?
Something. And are we talking about immortality? Not likely. Immortality, by definition, is never achievable, right? You never get there. But are we talking about eliminating aging? Yeah. I mean, I'll break the code of a gerontologist and say I think aging is inevitably going to be eliminated, and I think it will. I think in this century, people listening to this podcast many decades later in this century even will say, yeah, these guys saw all this coming. Yeah, the end of aging probably is in our century.
Amazing. And with what you guys are working on, the ability to induce tissue regeneration when things go bad, when you have injuries, when you have just things that pop up as you age. And then on top of that, what you guys are doing, the pluripotent stem cells, being able to just regrow organs that may go completely haywire. I'm inclined to believe what you're saying.
Well, I would hope so. I haven't said anything actually that's controversial in the scientists that are studying aging. I mean, there might be this or that one that would disagree, but that's this gulf of knowledge. The gerontologists I talk to, I mean, I do a reality check sometimes. I won't name names, but I ask some well-known gerontologists that are highly regarded and well-known, and they're in some cases competitors. That's a reality check. Do you agree with me? This is where we're at. I mean, yeah, of course. There's this gulf of knowledge. The gerontologists know all these things are real and are happening, and then the public isn't even aware of a lot of it. So, I would hope that people who are interested in this will just do a little research online and educate themselves further on it. It's fascinating history in the making. My role in it's just a part of it. There's many very dedicated and hardworking scientists. It's not just Mike West out there. There's a lot of hard work being done by a lot of people here, which we don't have time to name all these people.
Well, you've definitely been a massive part of it for sure. And I think that it speaks volumes that in the aging community and the people that are actually in the weeds that are working, it isn't a controversial thing to say that there is no potential limit to biological aging. And I think that speaks volumes. That statement alone speaks volumes.
If it were wear and tear, it'd be entropy, and that you'd say, well, it's a second law of thermodynamics. That's just part of nature. But we know that the second law does not apply to living organisms. Yeah, we just know that.
So the second question I wanted to ask you was, what is the most sci-fi-like thing you are excited about in the biotech industry right now? But I feel like our conversation has already gotten pretty sci-fi. Is there anything that is even another level above what we've talked about that's potentially on the horizon?
Yeah. Sci-fi when I think of sci-fi, I guess this isn't exactly sci-fi, but a dear friend of mine is Robin Cook. He wrote Coma, writes these medical thrillers, and he's a friend of mine. And I've asked Robin, why is it that the biotech scientist, whatever, is always the bad guy? I mean, maybe even intended good, but it always goes wrong, and they unleash some kind of Andromeda strain or some kind of monster or something goes terribly wrong. And he said, well, Mike, that's a formula. That makes great stories. When I think of sci-fi, I just naturally think for kind of the wild and crazy thing. And it's so easy, relatively easy, that is, to dream and use your imagination and to think how these technologies could be used to alleviate human suffering. For instance, just out of the hat, just give an example. HIV infection, AIDS. You probably heard of the Berlin patient. It was a patient that was treated with a bone marrow stem cell transplant, and it absolutely cured him. They can't find the virus anymore in his body. There's cells that can be transplanted into patients, and they'll actually be resistant to AIDS and destroy the virus. And we can imagine you could actually do this if you had the money and the resources. You could use these technologies we talked about today, make genetically engineered blood-forming cells that would be transplantable into an AIDS patient, or an HIV-infected patient, and they could rid the body of the virus and be cured of the disease. And just one example. But you could also imagine wild and crazy stuff. You could genetically engineer, soldiers with this double muscling phenotype, sort of an Aryan race. You could use it to make, there's a neurotransmitter that in animals, when you alter the gene, you increase the intelligence of the animal. Conceivably, you could tinker with human intelligence. As I mentioned, make dogs that glow in the dark. And you wouldn't have to put a collar on them when you take them for a walk at night. They would light up the street in front of you because they glowed in the dark. There's crazy stuff we could do. And that's where I had someone come to me once and I wanted to clone a dog and name him Glover, and a glowing dog. And I said, well, why? And he said, well, it'd be fun to take it to a party. This is the kind of frivolous thing where I worry that humanity would, with these immense powers that we're getting in science and medicine, where the sort of the science fiction, harebrained, maybe even intending well, but turning into something terrible, could go wrong. I think if we focus on alleviating human suffering, improving quality and length of life, I think these things are solidly in the human tradition. They're the compassionate use of science and technology. I think it's a compass needle I will follow all day long. But I'm certain that there will be people out there, for whatever motivation, will realize that they can do mischief with this new science. And it doesn't take a lot of imagination to think of different inventions that can be used. And many people will write novels and stories about how the science could be misused. So, yeah, but it's not all science fiction. I think some of it will become science reality, on the negative side as well as the positive side.
Well, you just got to hope for the goodness in humankind to make good use of this stuff. And obviously, there's a ton of people already working on that, including yourself.
Yep. For the listeners, everyone I know in medical research, 99.9% of them have very good intentions and are focused on life-threatening diseases that cause people to suffer, our loved ones to suffer, ourselves to suffer, and they should take some comfort in that. It's rare that I encounter the wild-eyed kook that wants to do something crazy. It's really quite rare.
Yeah, the mad scientist seems to be a trope that really maybe doesn't exist in reality that much, at least if they do.
It sells novels.
Yeah.
But yeah, it's just, I've been in the field, like this cloning field, that was really controversial. And everyone thought we were going to clone the Beatles and bring them back, and Lucy and Ricky and all kinds of stuff. But then there were a couple kind of fringe people talking about such things, but people are focused on medicine and doing good.
Super exciting stuff, Mike. I just have one more question. This is kind of maybe a personal question, and that I know a lot of my friends that'll be listening are curious about as well. What, if anything, can a 30-year-old male like myself do to slow down aging? Is it even worth trying right now and just waiting until stuff like what you guys are working on comes out?
No, I would just give the common wisdom on that. Don't smoke. Reasonable exercise regimen, moderation in all things, varied diet. I wouldn't. I don't want to provide medical advice, but I'm not a real fan. A lot of people are really downing a lot of supplements. I mean, I know some people that take 200 or 300 different supplements a day. I'm in this field. I study it in every waking hour. I don't have the confidence. I mean, a few of these are, you mentioned NAD and so on. There are a few of these I'm quite comfortable with, but most of these supplements we really don't know that much about. I remember years ago, the free radical theory of aging was the most popular theory of aging, and people were swallowing massive amounts of vitamin E, for instance. We now know that could have been detrimental. So I would argue a healthy lifestyle, varied diet, keep trimmed down, don't smoke, and then wait for technologies that are really going to make a difference by targeting the clockwork mechanisms of aging itself. At most, as you pointed out, based on animal data, you could hope to add a couple years to your life by such means anyway, and the risk of harming yourself through some of these supplements, it's an unknown variable, and oftentimes the unknown can come back and bite you.
What about the injection of stem cells?
Well, and I'm glad you brought that up. And that may be more candid here than is comfortable for me. When all the science was coming about and President Bush was talking to our country and everything, and this aging thing was mentioned, the quack medicine came out in droves. And without naming any company, if you see or hear that there's a stem cell therapy available today, come get them now. In my experience and in my opinion, the vast majority of those programs, companies, whatever they may be, are not based on evidence-based medicine, meaning there's no science behind it. So, if you're willing to ante up the money, they'll give you cells they call stem cells. And in all of them that I've looked at, again, I'm not making any judgment on any particular person or any company out there, but all of them I've looked at had absolutely, the ones that I've looked at, make it clear, have absolutely no science behind them. And in my opinion, they were quack medicine. So I would urge people that say, wow, that stem cell thing, it's really big, it's very powerful, and you see ads in newspapers, please be careful, because there's people out there who will take your money and they really don't care about the long-term consequences to your health.
And do you have advice for somebody maybe shopping around for these types of treatments on maybe quack flags, something that they can kind of, a red flag that will deter them?
Yeah, there are some. I don't have them handy. Maybe you could look them up and put them up on your website. I'll send some to you by email and you can put on your website. There are some resources. The National Institutes of Health has put up some information. There's some various resources out there to try to help people. But bottom line is the average person out there should know that 99% probably of groups that are reaching out and advertising are probably offering up unproven therapies that are not approved by FDA. So they're skirting FDA regulations and offering up unproven and potentially unsafe therapies. It's primarily driven by a desire for them to make money rather than actually help the patients. Sounds a little brutal, but it has to be said because it's, in my opinion, that's the state of the play at the moment.
Well, that it's a feeling I've had when it comes to stem cells, and I kind of just wanted to verify it with you. So that's some invaluable information.
But yeah, please be careful. The first cells ever isolated, pluripotent cells that started all this excitement, were called H1, H meaning human, 1 meaning the first line ever made. The first clinical trial was for spinal cord injury, and those cells are still, after now over 10 years, are still in clinical trials and are still not for sale. So when all these groups are out saying, we've got them now, buyer beware, please.
Mike, thank you so much for joining me today. I know you're such a busy guy, and this conversation honestly blew my mind. I think it's going to blow a lot of people's minds.
I've enjoyed it. Yeah. Long life and peace and long life to all your listeners. And please be safe now in the age of coronavirus.
If you got this far, I just want to say thank you so much for listening. If this was at all interesting to you, I'd love to connect on Instagram and hear your feedback. I'll also be posting clips from the latest episodes, as well as anything else I find interesting about the biotech industry. You can find me on Instagram at @SimpleBiotech. And if you're interested in the companies that I'm looking at and the companies that I'm excited about, connect with me on AngelList at angel.co/James Rule. That's James, Ruhle. Thank you so much and be safe out there.